at the National Museum of Natural History. Part of my work as a curator is to ensure that the national biological collections are in good preservation order. The primary role of the National Museum of Natural History is to take care, classify, and make available the natural history collections it holds for display, education, and most importantly, for research purposes. This is done in an effort to improve our understanding of the natural world as new characteristics to species definition are constantly being discovered. One of the collections under my care is the museum's fluid collection. In this masterclass, we will be looking at several aspects in the preparation and preservation of fluid preserved specimens, also popularly known as wet or embalmed specimens. Natural history museums in general are a repository for specimens of natural origin, which may vary from geological to paleontological and biological material. Varied in nature, these collections may be broadly divided into two different categories, according to the mode of preservation. These include the dry collections and the fluid preserved collections. Dry collections include specimens such as pressed plant materials, shells, skeletal material, pinned insects, mounted and study skins, amongst others. Fluid preserved collections, on the other hand, include those specimens stored in solutions. The usefulness of this preservation technique lies in the fact that in contrast to dry preservation, where only parts of the organism are generally preserved, such as skins or bones, in fluid preservation we're able to preserve the whole of the organism by submerging it in a liquid preservative that will provide protection against deterioration for long-term storage. This preservation technique facilitates the study of organisms' morphological features for taxonomic purposes. It also enables characterization and structure analysis of proteins as well as DNA sequencing, particularly for those organisms that cannot be stored as dried specimens. These include soft-bodied invertebrates, which require fluid preservation to support the body tissues and to prevent the shrinkage and distortion, such as jellyfish, for example, but not only. The most common biological material preserved in fluid include aquatic invertebrates such as echinoderms, mollusks, and crustaceans, and a number of vertebrate animals including reptiles and amphibians, fish, and also occasionally insects, micromammals, and plants. The idea of preserving organic material in fluids dates back to ancient times, where several fluids, including vinegar, brine, honey, and oil, have been used over centuries to preserve food, but not only. Long before the Egyptians perfected the mummification process, ancient Assyrians found a way to keep oxygen at bay to accomplish preservation, and this was carried out by embalming their dead in honey. In fact, it has been rumored that after his death in 323 BC, Alexander the Great was immersed in a golden sarcophagus brimming with honey. Egyptians have also been known to sometimes preserve important organs of the body in oil in canopic jars as part of the mummification process, including the stomach, intestines, lungs, and liver, all of which, it was believed, would be needed in the afterlife. Historically, a number of fluids have been used to store biological specimens, including brine, vinegar, turpentine, particularly prior to 1662. The Honorable Robert Boyle, who is considered one of the founders of modern chemistry and one of the pioneers in experimental scientific methods, was known to have experimented with brine solutions as a preservative, known as oil of spikes. Oil of spikes here refers to an essential oil made from lavender flowers. The English scientist Dr. Robert Hooke preserved leeches in vinegar, which was documented in his beautiful illustrated original work, Micrographia. Modern fluid preservation of biological specimens as we know it today began in 1662, with the discovery that alcohol could be used to preserve organic materials. The first recorded mention of the use of alcohol for fluid preservation was in fact on June 4, 1662, by the physician Dr. William Croone, who presented to the Royal Society of London 2 embryos of puppy dogs, which he kept for 8 days in spirit in a hermetically sealed glass vial. Boyle also experimented with fluid preservation and, based on his experience, recommended that the alcohol solution should be changed after initial preservation of the specimen, and that the preservative used should be redistilled, particularly when it became diluted or discolored. The testing of alcohol strength during this time used to be carried out by soaking strips of paper or cloth in solution and trying to light it up. If it failed to light, it was considered that the preservative was too dilute and requires changing. The idea of fluid preservation caught on quickly during this period, and some notable spirit collections from this time include those of Dutch natural scientist Jan Swammerdam, who was the founder of comparative anatomy and entomology. He used to preserve insects in spirits of wine or brandy. Much of his research on entomology was brought together by Herman Boerhaave in the book Biblia Naturae after his death, a book considered by some to be the foundation of our modern knowledge of structure, metamorphosis, and classification of insects. Another notable collection is that of Lavinus Vincent, a famous Dutch designer and merchant who used to collect natural specimens, amongst other things. His collection, which was made up of 8 cabinets, including around 600 vials of animal cadavers in spirit, is illustrated in the catalogue Elenchus Tabularium, which was published in 1719. And perhaps the most distinctive and preserved specimens from this time were those prepared by Friedrich Reis, a Dutch botanist and anatomist who is remembered for his groundbreaking methods of anatomical preservation. Reis' chief skill was the preparation and preservation of specimens, using a secret solution known as liquor balsamicum, in an effort to keep scarce and expensive cadavers around for longer period of time by wet preservation. He is well renowned for his impressive collection and bound specimens, which intended to serve as an educational tool as well as display for his artistic expression through the creation of intricately decorated jars in which he kept specimens. Another significant natural history collection containing fluid-preserved specimens from this period was that of Dutch apothecary Albertus Siba. Siba was known to have purchased several of his specimens from sailors at docks and from commercial dealers. Initially, fluid preservation was a laboratory procedure. However, it quickly became a means for preserving specimens in the field. By the early 1800s, the published recommendations for fluid preservation used to specify the use of beverage alcohol or spirits of wine with various chemical additives, including acids, glycerin, salts, arsenic, ammonium chloride, alum, and others. At the time, rack, rum, and brandy were also used as preservatives during the period, especially on voyages of discovery. In fact, the natural historian and collector James Petiver has published a sheet of instructions called Brief Directions for the easy making and preserving collections of all natural curiosities for James Petiver, Fellow of the Royal Society of London. This sheet indicated that all small animals such as beasts, birds, fish, serpents, lizards, and other fleshy bodies may be preserved in rec, which is an alcoholic beverage distilled from the juice of coconut palm, or a mash of rum and molasses, rum, brandy, or any other spirit found on ships. When these were unavailable, then it was suggested that they are to be preserved in brine, or seawater, or even pickled. The preservation of specimens in fluid made it also possible to create more accurate illustrations, such as those produced by the German-born naturalist and nature artist Maria Sibylla Merian. In one of her letters to her friend Dr. Johann Georg Volkamer, she described her experience in Suriname, and she wrote, "When I was in that country, I painted and described the larvae of caterpillars, as well as the kind of food and habits. Everything I did not need to paint, I brought with me, such as butterflies and beetles, and everything I could steep in brandy." In 1859, formaldehyde was discovered by the Russian chemist Alexander Butrilov, and later identified by August William von Hofmann. Its true value as an antiseptic was demonstrated by the German physician and biologist Ferdinand Bloom and others between 1892–93, and it was found to be a better primary fixative than spirit. Bloom showed that formaldehyde was more effective at maintaining the color and shape of biological tissues without affecting the structure of specimens. However, formaldehyde has since been found to be unsuitable for preserving DNA. For this reason, it was later recommended that tissue should be fixed in 10% formaldehyde and then transferred to alcohol to bring back the color. This two-stage process of formalin fixation, followed by alcoholic preservation, forms the basis of modern preservation of fluid-preserved specimens. At present, the National Museum of Natural History holds more than 700 preserved specimens in fluid, some of which date back to the early 1950s. The majority of the preserved material is from the Maltese Islands and is made up of organisms belonging to several classes, including hydrozoans, crustaceans, echinoderms, annelids, herpetological material, fish, and mammals, particularly micromammals, including bats, shrews, and rodents. Type specimen refers to that specific animal or plant that was used by taxonomists to describe a new species based on the characteristics of that particular specimen. The details are published in scientifically recognized journals. Hence, the published scientific name and official description, which defines and characterizes that particular species, will be then permanently associated with this type specimen. Voucher specimens, on the other hand, are specimens collected as a representative sample that has been used for identification and as a supporting evidence of information learned during a particular research project. Therefore, based on this, it is critical that these specimens are preserved correctly so that the information they hold is available for long periods of time. Of particular interest is the echinoderm collection donated by one of the museum's volunteers, which contains a number of type and voucher specimens. This particular collection is stored in absolute ethanol as a reference collection, since this preservative will not interfere with the molecular structure of DNA. The preservation of fresh biological material involves four main stages: documentation, fixation, staging and container preparation, followed by preservation. The fixation and preservation of a specimen strongly influences the future availability for study. Therefore, it is important to determine whether the specimen is to be preserved as a research resource or for another purpose, such as display. Most of the time, specimens are acquired by the museum either through donations, confiscations, as part of research studies, or monitoring programs. In an effort to enhance the value of these specimens, we strive hard to make use of good preservation practices and documentation, since these all enhance the value of the collection. As part of the preservation technique, the primary stage is a thorough documentation of the specimen. In many cases, specimens are frozen to halt decay as much as possible. Hence, the first step in preparing fluid-preserved specimens is to actually thaw out the specimen completely and proceed with the documentation. Thawing is carried out by placing the specimen on a tray covered with absorbent tissues so that any leakages can be disposed of easily. The specimen itself is also usually covered with paper towels and checked periodically. Each specimen is identified and, where possible, sexed, aged, measured, and photographed. Finally, a specimen number is assigned to each individual. The next step is to fix the specimen. There are a variety of fixatives available for preserving biological material. However, the selection of the fixative is usually based on the intended use of the specimen and the efficacy of the fixative. The primary role of the fixative is to halt and prevent any post-mortem changes in the specimen whilst actively preserving it. In practice, there are two main types of fixatives: aldehydes and alcohols. The most common fixative used to date is formalin solution, which is made up by mixing formaldehyde and water in a predetermined concentration. An alcohol can be considered as a fixative because it is able to change the protein structure and promote the hydration of tissues. In fact, until the 1890s, alcohol was used as a general fixative. At 95%, alcohol is able to fix DNA and is also suitable for long-term storage. The exposure of biological specimens to aldehydes in general denatures DNA. Therefore, in such cases where the type specimens are involved, the fixative used is ethanol. Other less commonly used fixatives include heavy metal fixatives such as mercury chloride and osmium tetroxide. These are, however, used for small specimens and tissues. The general procedure for fixing a biological specimen is achieved by injecting it with a fixative that is able to stop the natural deterioration and decay process, also known in biological terms as autolysis. This is either carried out by using 10% formalin, alternatively by using directly 70% ethanol or industrial methylated spirit. At this concentration, ethanol becomes an effective biocide. For fixation, the concentration is an important factor, since if higher percentages of ethanol are used, there will be specimen dehydration risks, and if a lower concentration is used, it will not be an effective biocide. Prior to immersing the whole specimen in the fixative solution, the organism is often injected in various locations with the fixative. Special attention is given to a number of areas depending on the specimen type, including the head, neck, abdominal cavity, legs, and tail. Perfusion of the fixative into the tissues on large-sized organisms is ideally carried out by injections through the specimen's circulatory system. During injection, the specimen will start to appear bloated, and fluid may start to leak out, which indicates that the area is saturated with the fixative. Whilst injecting specimens, it is very important to maintain distance between the hand and the needle, so as not to end up poking oneself and end up being embalmed instead of the specimen. Historically, before the availability of reliable syringes, fixation used to be carried out by making cuts through the specimen to allow for the penetration of fixatives. Fixation causes many of the tissues to tighten and harden, and therefore, prior to immersing it in a fixative solution, the container in which the specimen will be stored and the positioning of the specimen in the jar are prepared. Otherwise, it will be very difficult to move, bend, or reposition the specimen following fixation without risking damaging the specimen itself. For display purposes, specimens are often staged onto a glass backing plate. The specimen is then placed on the glass plate and strategic points are marked, where holes are to be drilled to be able to thread mount the specimen on the glass backing plate. After drilling holes in the glass plate, the specimen is tied securely on the backing plate using thread and submerged in the fixative solution to fix completely. The specimen is then left for a period of time in the fixative solution, depending on the size and type of organism. This step is an important part in fluid preservation because it ensures the effective fixation of the entire specimen from the inside out. After a day or two in the fixative, particularly if formalin solution is used, the solution is changed to a buffered formal solution. The addition of buffers ensures that the pH of the fixative remains stable and does not become too acidic. A label with the specimen name, specimen number, fixation date, and person performing the procedure is added to the container with the specimen for record purposes. There are many types and sizes of museum jars available today, including Kilner jars, Beta jars, Visi jars, Bakelite lidded jars, gasket jars, etc. Moreover, larger specimens may also be housed in custom-built tanks. Following fixation, the specimen is then transferred to a stable storage medium for long-term preservation. The meaning of the term preservation in this case refers to the maintenance of a specimen in a stable and usable condition, and the chemical chosen must ascertain that the rate of deterioration is reduced to a minimum. For a liquid to be considered a storage medium for preservation, it must be able to hinder biochemical reactions from occurring, prevent any internal and external microbial attack, provide structural support to the specimen to preserve its three-dimensional structure, and maintain the specimen in as near a natural state as possible. Common preservatives used as storage media include ethanol, isopropyl alcohol, glycerin, and glycol, formaldehyde or glutaraldehyde, or oils, for example, mineral oil. The transition from fixative solution to a preservative solution takes place in a series of increasing ethanolic concentrations, from 20% up to 75%. Specimens in collections should always be labeled with the species scientific name, the name of the person who found it, cause of death, the date of collection, specimen number, and the name of the person who preserved it. Containers should always be labeled to indicate which fluid is inside. The data is digitized and stored in a database. Internal specimen labels are usually used, since these are not susceptible to pest damage and are easily transferred between containers with the specimen. This, however, requires the label to be made out of a material that is able to withstand the aqueous conditions. Generally, 100% cotton stock paper or synthetic label materials made out of span-bonded polyethylene are used. Currently, at the National Museum of Natural History, we are using resistal paper. Traditionally, information was written in black Indian ink, although alcohol-proof inks have recently been developed and are now being used as well. Preservation jars can be sealed with different types of closures, including snap-on lids, screw-on lids, gaskets, and ground glass. For this particular jar, a ground glass seal will be used. The glass jar is kept 3/4 full with the preservative, whilst making sure that the specimen is fully submerged in fluid. The top edge of the jar and the lower edges of the lids are then ground. Lid grinding is essential, otherwise the sealant will not be able to attach properly to the container and the lid. Grinding is carried out using a carborundum stone or sandseed, aided by some water mixed with glycerin. Any sharp edges on the lid are removed and then the stone is passed over the lid surface in circular motion until the glass surface is sufficiently ground. Several methods exist for sealing glass jars. However, a tried and tested method for sealants is using gelatin as a sealant. This procedure was discovered in an Index Museum daybook dating back to the mid-19th century at the Natural History Museum in London. This method is still used today with good gelatin seals lasting for more than 20 years, and at the National Museum of Natural History we usually employ this technique. Is topped up with alcohol to 3 or 5 millimeters below the lid. The prepared sealant is then melted in a hot water bath, about 80 degrees Celsius. The sealant is then brushed fairly generously on the outer edges of the jar, and the glass lid is then placed on the applied gelatin seal. The jar is then checked and further gelatin is brushed if necessary, to fill any gaps that may be present. Weights are then applied to the lid and it is left to dry overnight. The jar is then inspected for white streamers of excess gelatin, and if the appearance is satisfactory, it is placed in a cabinet as part of the reference collection. Our mission at Heritage Malta is to ensure that those elements of cultural and natural heritage entrusted to us are protected and made accessible to the public. Here, at the National Museum of Natural History, we are committed to the safeguarding and preservation of natural history treasures within our care for future generations. The Fluid Preserved collection is just one of the ways we live up to our motto of ensuring a future to our past. I am Stephanie Sammut for Heritage Malta. Thank you for viewing.